Welcome as well to the panel this evening, Professor Patricia Duffy Barber, who is our Vice Chairperson here in Cystic Fibrosis Ireland and is also a person living with CF. So welcome this evening.
Hi, Sarah. Thank you. I'm not a professor, but it did kind of have a nice ring to it, didn't it?
Well, a professor of life experience with CF. So I think it definitely qualifies you this evening. Yeah.
Thanks.
So, yeah.
I was just wondering, I did have a quick question. I don't know if I can squeeze in before questions from the viewers. I suppose just really, I was kind of wondering, like, how would I know if the person with CF in my family who has a rare mutation, how would I know if that's already been looked at as part of the biobanking? Just be curious to find out.
Well, I think that the project is organized via the CF centers. So the CF center of the patient and the treating physician should be aware whether a patient is biopsy yes or no. Well, if there are specific questions, you can always reach out to the HCF organoid organization via the website. But in essence, the treating physician should know whether a patient is biopsied, yes or no.
Thank you, Cors. You mentioned that you are still including new patients, or you're hoping to include new patients in that biobank. If there was people in Ireland who were interested in having their organoids tested, is that something that they could sort of refer to, or what is the process by which you recruit people to your study?
Yeah, well, during the Corona period, we stopped with collecting the biopsies, but we really want to start it up again after, well, let's say the summer or so. But there are still some issues with funding, so we have applied for new grants at the European Commission. And in some countries, patient organizations are very helpful to support the project. So as soon as there is funding, and I hope that it will be after the summer, that we can reach out to centers again to start collecting additional biopsies from patients with rare mutation.
That's great news. If the HCF shows proof of concept that organoid testing, that model, is a marker for success of a treatment, how long do you think before organoid testing might become a regular part of the personalized approach to CF care?
Well, that's an important question, of course, because if it's acknowledged as a test, and what we see in the US is that the FDA has already approved label extension for example, Kaftrio based on cellular models, but it's not the case in Europe until now. We are currently discussing with EMA to acknowledge the organoid test as a test for CFTR. So we have asked for a qualification opinion. That's a sort of formal process in which EMA can approve the organoid testing as a measure of CFTR, but also as a predictor of disease severity and also as a predictor of therapy response. So it's currently under debate with the EMA, and we hope that, well, within some 3 months from now, we have a formal answer from EMA whether they can approve this test as a formal test for CF. So that would be a major step forward for all of us.
Yeah, absolutely. We certainly look forward to that outcome. Professor Harrison, I might come back to you. You mentioned that some of the early stage trials in nasal epithelial cells might be happening next year. And I was wondering if it's shown to work, these are the basic editing mechanisms. If it's shown to work in nasal epithelial cells, which presumably respond or is similar then to the respiratory tract, could the same mechanism then be used to restore gene function in organs like the pancreas or the liver?
Yeah, so one of the data slides I showed there with our collaborators in Johns Hopkins, when we isolate, or when they isolate nasal cells, we can base edit those very efficiently. So the question now is, there's a few safety things we need to have a look at, and then see if we can set up that trial to just do, without being too rude, just inside the nasal epithelial there, just applying a small amount of this gene editing medicine and you do the experiments on one side and a control on the other side, and then maybe come back and have a look at that a week later and see, you know, if the cells are still there, if they've been damaged, if it's been effective. In terms of then going to other tissues, which was your question, like the pancreas or the liver, I think the overwhelming majority of researchers, and also I've got this from people with CF as well, is to focus on the lung. Let's see if we can sort out the lung first and then start to look at other organs. But that's also driven in some way by the way that most people assume that this will be delivered and that it will be inhaled. But another completely viable option is to do it intravenously. So if we go for an intravenous approach, what will happen is the majority of the editing will occur in the liver. Just by default, that's where the circulatory system takes most gene therapy products. And the example which I've shown with the clinical trial from last year, people initially have started looking at liver diseases just because they know if you inject something intravenously, that's where it will go. So the liver with CF is very important. It's not the priority in the field, but I think when people start to look at delivery methods and they go through an intravenous route, then that's something that people will start to look at along with the pancreas and along with the digestive system as well.
Thank you. You mentioned there some of the delivery methods, and that comes to another question we've just had. How would gene editing be administered? So I know you mentioned there IV and applying it to the cells inside the nose. Are there other mechanisms of how by which gene editing would be administered?
Yeah, so they're the main two routes which you've got. It's, you know, you've got a layer of epithelial cells in the lung, so you can either come directly into the lung and try and get them that way, or the circulatory system, which is in very close proximity on the other side. So they're the two routes. In terms of the chemicals or the reagents to deliver that, broadly speaking, there's two mechanisms. People talk about viral vectors and people talk about non-viral vectors. So we've all been, or the vast majority of us have been injected with one of those in the last 2 years. So if you've had the Moderna vaccine, that's an mRNA molecule as a lipid nanoparticle. So the formulation for that would be very similar to what you might envisage for one route. Or if you had the AstraZeneca vaccine, that was a virus vector expressing a protein, which is what you'd want as a delivery mechanism for this. So the fact that they've been developed and developed quickly, they've been used in literally billions of people with an extremely good safety profile, bodes well for delivery methods in people with cystic fibrosis. The challenge is just literally the size of the CF, the size of our lungs anyway. You know, it's roughly the area of a tennis court, so you need to deliver a massive amount of the virus vectors. Also in the CF lung, you've got several factors fighting against gene delivery. You've got lots of thick mucus, which you need to penetrate through, but there's certain mechanisms which can get through that to some extent. You've also got the immune response, antibodies and an inflammatory response, which is trying to fight those infections. So you're trying to get into somewhere which is actively trying to keep you out. So there's a lot of challenges to solve. I think from the development of the editing strategies, you know, if this is where we started, that's where we've got to be. We've come quite a long way. With delivery, we're getting there, but there's a gap to catch up with on the delivery.
And that leads quite nicely into the next question, I suppose, which is when you do deliver them, do they need to be applied to germ cells or could they be applied to regular somatic epithelial cells in the lungs?
Depends who you speak to. Nobody knows. Nobody knows because we've not got it to succeed. Most people in the field are quite adamant that it needs to be the lung stem cells if we're focusing on lung. And the reason for that, which makes sense, at least in principle, is that those cells will be providing new epithelial cells. That's where all our epithelial cells which line our lung have come from. The possible counter argument is that in the lung cells may not turn over that quickly. What I mean by that is that the basal cells or the stem cells don't produce a new epithelial layer unless the lung is damaged. So if large proportions of lung cells aren't being damaged, and you were very successful at editing the basal cells, you'd have a basal cell edited and ready to go. but until it produces some new cells, they wouldn't actually be functional. So if there wasn't actually lung damage, then these basal cells could be very highly edited, but then don't actually proliferate and do the function that you want. So one argument is that maybe the surface epithelial cells would be a better target. They may be easier to reach in the first instance, but until we do it, we don't know. But one of our projects funded by the CF Foundation just at the moment is to try and investigate this. So I won't go into too many details, but we have a lung cell line with a group in Portugal. They're actually at the conference just at the moment. And they can differentiate those cells into all the different lung cell types. And what we want to be able to do is to set up our editing so we can edit at different stages of development and then ask the question, what happens if you edit just a stem cell? What happens if you edit just a club cell or whatever? And at different time points, what effect does that have when we grow these complex epithelial layers in a dish, which may give us some answers towards that question? I think ultimately, let's just see what we can do rather than get too specific about what we want, because we don't quite know yet.
Thank you. Professor van der Ent, I might come to you with the next question. Asked, will a person who had one copy of the delta gene and a rare second gene be included in these rare studies, or is it thought that Kaftrio would be most suited for them?
Yeah, well, currently the Kaftrio is available for both patients with one and with two delta F mutations. So all these patients with homozygous and heterozygous delta F mutations are not incorporated in the HCF project. So we are really focusing on the ultra rare mutations. So also all the heterozygous patients with delta F mutation can be treated with Kaftrio quite effectively. Yeah.
Mm-hmm. Could the biobanked organoids be used to test the likes of gene therapies, the amplifiers and the correctors, or is it just the modulators?
No, essentially we could test all the different types of therapy, and Patrick told us a lot about gene therapy, and for example the MRT 5005, it was already tested on the organoid. So we can also do gene therapy on organoids, and we also described it already. And I think that one of the questions that was raised by Patrick, so how can we bring the compounds into the cells, can also be answered in the organoid model of individual patients, because of course you have the inhalation, and where does it come into the lung, and is it trapped into the mucus? But the question is always, does it also enter the cells? And with the organoid models, we have the models in which we can bring in the compounds quite directly, so that we can at least test whether these therapies are effective if we are successful to bring them downside into the airways and lungs. So I think it's also a very interesting technique to use for gene therapy, and we have been working with the intestinal models in the HCF project. But in the meantime, we have also developed the lung model and the nasal model. So there are also nasal organoids and pulmonary organoids available that we can use in the same manner as the intestinal ones. And that was the attractive thing in the high throughput screening of the huge amounts of patients, is that the intestinal model is quite fast. So they are replicating within some days. And for the pulmonary organoids, it takes much longer time to grow them and to study them. So, but it's possible to do the same type of studies in personalized cell models from individual patients in respiratory epithelial cells.
And are the responses in the cells taken from the respiratory tract and the gastrointestinal tract, did they respond differently with it from the same person?
Yeah, more or less, in that sense that in the intestine, the swelling response is fully CFTR dependent, while in the respiratory epithelial, there are also other chloride channels that can be influenced by your compounds. So alternative chlorides that are present in the respiratory epithelial cells can also be well, influenced by compounds, and so that causes sometimes some differences. But if you look to the CFTR function itself, then the most pure model is the intestinal model.
Thank you.
Just a quick comment there. It's slightly different, but I think it's relevant. One of the things that we've just started to notice over the last couple of months, even with the base editing, is that we get slightly different responses in different cell types. And there's some off-target effects or bystander effects, and they're different in different cell types. And in, you know, your colleagues, Jeff Beekman and Hans Kleber, they've done an editing. They got a slightly different profile to us in one of our experiments. And then we've seen it now in a different cell type again, that we get something more similar to them. So I think there's a lot of subtleties that we need to work through.
Sure.
It's nice to see its uniqueness even within the same person. CF has lots of unexpected twists and turns. Patricia, I might bring you back in here. Have you any additional questions or comments?
Not really a question, just more of a comment of just, you know, sitting back and looking at both of the talks, and just, it's so reassuring to hear that there's so much constant research going on, and it doesn't always make the headlines until there's something that's a bit exciting, but that never give up, I think, was really reassuring that, you know, sometimes, you know, research is really more the tortoise than the hare. And, you know, it can be the unexpected findings that lead gradually towards something really exciting. So I think really, yeah, it's just expressing that sense of just really reassured that people are still working away. And we really want everybody to have something that is really meaningful and makes such a big change as the modulators have for so many of us. And I suppose as well, we're very conscious, you know, economically in this country, we have access to the drugs, but that very much is a different story across the world. And it's about hoping that everybody really can access something that works for them in a, you know, financially sustainable kind of way. That was just what came across. Thanks.
Thank you, Patricia. And I think that Amelia Earhart quote is quite relevant here, and it's great to see so many people working on so many unmet needs in the field. I would like to thank both of our speakers. Thank you very much for joining us this evening. We could go on asking questions all night, but thank you for giving up your time. I hope you enjoyed the rest of the conference in Portugal. Patrick and Professor van der Ent, we hope to hear more about the HIT-CF project over the coming months. So thank you both very much. Bye. Patricia, I might hand back to you just for the final closing message. Thank you.